we are getting you right, sir.
No, what I'm saying, am I not saying more than what I'm supposed to say? Because the time is taking much time.
No, sir, we are understanding actually.
No, sir, we are understanding you.
Okay, so we should continue
The way we are doing, man.
we are doing. Okay.
Yes, sir.
So now we have this membrane, ability to form the membrane. So for example, if by any chance, now trying to explain what I call the self-healing. By any chance now you disrupt, for example, this micelle, you disrupt this by removing or pinching this and remove some of those phospholipids. What will happen? They will come back together and heal themselves. Because they automatically form, because they don't want to interact with water. So if you touch them or you remove some of them, the remaining ones will come back and heal themselves. That is what we call the self-healing. So this is how these phospholipids behave inside water. So automatically when you put them inside the water, when they are in enough concentration, they can actually form either a micelle or this liposome. This liposome has been used in the medical line, most especially in drug delivery. When you want to deliver a drug to a particular organ, we'll talk about some of this, the basic knowledge. You will know what they are. And you can actually, for example, here, you know, some of these drugs we are taking, quite a number, majority of them, they are more of lipid soluble than water soluble, because the body doesn't take much of this or absorb much of this water soluble, highly water soluble. When you take a drug that is highly water soluble, you discover the bioavailability is very low. That is why you will see some difference with respect to the concentration when you are taking a drug. This one is 2 milligrams, while another one you have to take 200 milligrams, another one you have to take 300 milligrams. In most cases, those differences are actually depending on the bioavailability. How easy can that drug be absorbed? If that drug can be absorbed, enough can be absorbed, then you don't need much. But if, for example, it is highly water soluble, you find it very difficult when you ingest it inside your stomach for it to be absorbed by the membrane to enter into the cells. It is very difficult. So maybe out of 300, you may end up having 10 or 20 milligrams being absorbed. So you can see you cannot take just 20 milligrams. When you take 20 milligrams, you may end up with only 1 milligram. And the 1 milligram may not do what it's supposed to do inside your system. But when the drug is water soluble, you can take 20 milligrams, and there is high chance that all the 20 milligrams can even be taken up by the body. And this is through the membrane transportation. We talked about membrane transportation in some of the study session. So this liposome has been used in area of medicine for drug delivery, for example. Some drugs can be encapsulated here inside, and when this enter your body, they can easily move inside the cell through the process we call endocytosis. We talked about endocytosis and the exocytosis. So they can easily get into the system. And in addition also, it has also been used in a particular cell targeting. For example, when you are given a drug and you want that drug to move to a particular type of cell, for example, cancer cells, you don't want this drug to go anywhere, because normally the drugs, the way we take drugs, it enter the body, it enter into the blood, and the blood will pump it to different types of cells. So when some of those will get into the area where we have this problem, that is where they solve the problem. But there are other cells that this drug can go there, but they are not faulty or they don't have a disease. So those cells, those drugs does not do anything there. But when you are just targeting that particular cell, but see, this my drug will not enter any cell, will not go anywhere but the cell where this problem happened. You may decide to target that cell using this liposome. We talked about glycoproteins. We'll see how these glycoproteins can be added here inside this, so that when the glycoproteins get into the cell can recognize them that, oh, this thing I can easily recognize some glycoprotein on the surface of the cell, so that they can attach themselves. And as such, by attaching the process of endocytosis, where this content can be emptied inside those cancerous cells or whatever cells you are targeting. This is a classical of what we call liposomes, and how this behavior of phospholipid in aqueous solution can lead to formation of a phospholipid bilayer, as we've seen in a membrane bilayer. And that is the reason how or why the membrane form a bilayer. Sorry, I don't know what happened. I cannot move to the next slide. I don't know. Is someone hijacking this, and I cannot move to the next slide. Let me stop the sharing and share again, please. So the next thing we talked about is asymmetry of lipid bilayer. What is asymmetry of lipid bilayer? We talked about a different type of lipids, phospholipids in particular, and we said we have seen how this lipid form two layers. And we talked about a different type of phospholipid. So actually, this lipid is found within these layers. You can find the different type of phospholipid in either of the layer, the inner layer or the outer layer. This is what we call the outer layer. This is the inner layer. So different type of phospholipid are most commonly found. Some of them are found in the outer layer, while some are found in the inner layer. For example, in red blood cells, you have a phosphatidyl choline and sphingomyelin, mostly found in the outer monolayer, whereas the phosphatidylserine and phosphatidylethanolamine and maybe some glycolipids and glycoproteins are found in the inner layer. Are we together?
Yes, sir.
In most cases, and this is specifically to red blood cells. So you can have some situation where they can change in position. For example, one cell undergo apoptosis. What is apoptosis? Apoptosis is a programmed cell death. We just talked about lifespan of a cell, that after some time the cell will have to die. How does the cell die? How do you know that this cell is dead? Something must happen to tell the body that, see, this cell is no longer alive, or this cell reached its own lifespan. And how does that happen? Now, based on the composition of this phospholipid between the inner and the outer layer, when you have a cell that undergone apoptosis, phosphatidylserine that is usually located in the inner layer can now translocate, can now move to outer layer. For example, we are now seeing an inner layer here. You have phosphatidylserine. In most cases, you have phosphatidylserine. So this phosphatidylserine and phosphatidylethanolamin are mostly found here, but not commonly seen in the outer monolayer, but in the inner monolayer. When the cell is dead, you have translocation of phosphatidylserine and it will move to here. So when it moves to here, it is a signal. It tells the other part of the body, see, this cell is dead. We are receiving a signal. The signal is through the movement of this phosphatidylserine from the inner to the outer layer. So it can easily be recognized, and then it can be targeted for killing. So the cell can easily be taken into the lysosome and then break down to the other components so that the other component can be utilized, or new cell can be used, can be produced from that. So the next thing among the membrane lipid is the glycolipid. We talked about phospholipid. So just like a phospholipid, in the case of glycolipid, what we have is the sphingosine backbone rather than the glycerol backbone. So just assume the fatty acid attached to the sphingosine instead of the glycerol. In the third carbon, instead of you have phosphate are now attached to either choline, ethanolamine, inositol, and the rest. In the case of glycolipid, we have a sugar attached to it. 1O2 sugar moiety give what we call cerebral soil. Or you can have more than 2 sugar to form what we call a ganglioside. You can see a membrane here. On that membrane, you can see a lipid here. But on that lipid, you have attached to it a glycolipid, sorry. So you can see this as an example of a glycolipid. So we also have cholesterol. So in between this phospholipid, glycolipid, you can also have cholesterol. And cholesterol, when you look at the structure, the cholesterol contains some aromatic rings and an OH group. That OH group is the polar part of the compound. That polar part of the compound is what now interacts with the hydrophilic part. And then the aromatic ring, which is purely hydrophobic, will now interact with the fatty acids. Cholesterol is amphipathic in nature, and it also helps in stabilizing the membrane by increasing the membrane fluidity. Remember, I talked about 2 characteristics of membrane. I talked about membrane being self-healing, and I explained why it is self-healing. I talked about the membrane being fluidy in nature, not rigid, and we gave an example, a reason why they are not rigid. We made a mention of the fatty acid with regard to saturation level of the fatty acid, or whether the fatty acid is long or short, and how that affects the fluidity of the membrane. So also, you have this cholesterol. The cholesterol also helps in increasing the fluidity of the membrane. Thereby, the membrane will not be rigid. It is not good for a membrane to be very rigid. If it is very rigid, it will not allow, in fact, it will not allow movement of substance in and out. It will make the cell easily die because nothing will go in, nothing will go out, and it will not be flexible. There are so many processes to take place, which we will see some of this process that relate to a cell membrane. Now, another thing is the membrane proteins. Also, in addition, you can also find proteins inside the membrane. These proteins are divided into integral proteins and peripheral proteins. Peripheral proteins are the ones that are attached to the outer layer basically attached to most of these the hydrophilic part of it. Or you have the integral membrane protein. The integral membrane proteins are the ones that transverse from the inner layer to the outer layer. So outer and the outer side or the outside, they interact with the polar head groove. And the other side, inside also they interact. But in between, they interact with the fatty acid, which is purely hydrophobic in nature. So in most cases, we talked about fatty acid, we will talk about amino acid. And we also see the different type of amino acid. Some of these amino acids are hydrophobic in nature also. So in most cases, when you have an integral membrane protein, you will find out that some of those amino acids that are found around this area where it interacts with the hydrophobic part of this membrane are usually a non-polar amino acids. They are hydrophobic amino acid also. So you can see the hydrophobic only interact with the hydrophobic. For those ones that are found around the hydrophilic region, they are usually hydrophilic also in nature. So this is how the interaction happen, and this is how this thing is made possible, that you can have a protein that transverse in between the two layers, one being hydrophobic while the other being hydrophilic in nature. So basically, this membrane protein can serve as a receptor for hormones. They can serve as enzymes. They can serve as transporter that allow the movement of substance, just like this one. You can see this one form a kind of a channel where substance from outside can get into inside of the cell. While some like peripheral, we can see peripheral is only attached here, but it does not transverse. So they can also serve as recognition or adhesion molecules like glycoproteins. We'll give an example of glycoproteins shortly. So that we said, they can serve as recognition molecule called glycoproteins. Now let's give a typical example of something we all know. Blood grouping. Why do you have a blood group A or blood group B or blood group AB or blood group B? It is simply due to these glycoproteins that are found inside the membrane. What are these glycoproteins? They are membrane proteins that are attached to sugars or attached to carbohydrate. Now, we have this type of membrane glycoprotein that is used to determine the blood group of an individual. So different individuals can have, some can have O, some can have A, some can have B. Depending on what type of glycoprotein is expressed on the surface of your red blood cell. Here we are talking about red blood cell only also. So you have a red blood cell. Attached to that red blood cell surface, you have a glycoprotein. And attached to that glycoprotein, for example, let me give this protein. Attached to that protein, you have some carbohydrate attached to it. Let me pick this example, this one. You can see it, you can see some carbohydrate attached to it. So it serves as a recognition. So when you have an individual, for example, who is an O blood group, that individual will have the glycoprotein. You can see the red blood cell here, the first one. And on the red blood cell, you have the protein on the membrane. On that membrane, you have some sugars attached to it. You have the first one, which is yellow. We can see yellow, is galactose. We can see the key here. You can see the keys here. So the first one is yellow, which is galactose. The second one is blue, which is N-acetylglucosamine. The third one is yellow round, is still galactose. The fourth one is red, and it is triangle, which is glucose. So when you have an individual with a cell that contains these sugars, that individual is blood group O. That individual is blood group O. You can also have another individual with exactly like this, but on this galactose, you have another sugar, blue, but square, and this is N-acetyl galactosamine. So that individual is blood group A. You can also have another individual with exactly like that of O, but still on that galactose, you have another galactose attached to it. That individual is said to be blood group B. Now you have another individual that have something that exactly look like A and something that exactly look like B. That individual is classified as blood group AB. Class, are we clear? Did I make this explanation clear?
Yes, sir. Very clear, sir.
Yes, sir.
Okay, please, do I have a question?
So I have a question, sir.
Please go ahead, make it fast time, please.
So when you are talking about those lipid chains,
Sorry?
so can you hear me?
Yes.
When you were talking about the lipid chains, those unsaturated lipid and saturated lipid, I want you to throw more light there, sir.
I didn't talk about change. What I said is that sometimes you can have those fatty acids. Some of them can be saturated, some can be unsaturated. So the level of saturation, determines validity. If a fatty acid is saturated, there is no possibility of forming what I call kink bond, that bending. So if it doesn't bend, meaning the fatty acid can pack together. But when they bend, they can disrupt the packing. So disrupting the packing is what increases the fluidity of the membrane. But if they do not have this double bond, there is no bending, then they can easily pack together. And the more they pack together, the more they become rigid, and the more the membrane becomes rigid. Is that clear?
Yes, sir. Thank you, sir.
Yes. Another question.
Right, sir. My question is on the blood grouping you use as an example here. Before now, I used to know that is the presence or absence of an antigen on the surface of the red blood cell that determine the blood grouping.
Yes.
But I guess I'm learning. But I think your explanation, I don't know if it's same or in line with the absence or presence of antigen
Okay. So
on the surface of red blood cell as a determinant of blood grouping.
If I may ask you a question, then what is an antigen?
Okay.
You get me? So
yes,
the thing is, you are not wrong when you say presence or absence of antigen. You are not wrong. What is an antigen is a question. So you have to understand what is an antigen. Here in this case, we have this glycoprotein as the antigen. We are talking about biochemistry. You know, in biochemistry, we will not just tell you antigen. What is that antigen? We have to tell you exactly what it is. If it is a biochemistry student, we'll have to even show them. They will know the structure of those the elements. They will know the structure of galactose. They will know the structure of all the N-acetyl glucosamine. They can write it in the chemical format. Did you get it?
All right.
Yeah, so
yes,
at the theological level, we just talked about antigens without more detail on what are those antigens, what exactly they are. But
Art
here we
teachings.
are telling you this is exactly what they are. And when we go deeper, we
All right,
talk
sir.
about the chemistry, and to some extent, you will, when we say galactose, you can even remember the structure of galactose, chemical structure. When we say N-acetyl glucose, you can see the structure in your head, the chemical structure of N-acetyl glucose.
Section. All right, sir.
Do you have any other question? So in the absence of question, and this part also, I have this question. So this question will be posted on the LMS. You go and answer those questions as a quiz. Please try and do that. I will do that maybe before the end of today. And another thing also, I've recorded this class, like I told you. I will upload it to a YouTube channel, and I will share the link through the WhatsApp group. So anytime you feel you've forgotten one or two things, you can easily refer back to them. I used to keep, in fact, even the last year classes were there. So if you feel that maybe this one didn't explain something enough, you can look at any corresponding tutorial or corresponding class we did last year and watch over, and you can be able to grasp. I am very sure that with this little explanation, if you actually attend the class, you will have a clear image of what this study session 1 and 2 encompasses. And if that is not clear for you, you can have it there. The reason why I decide not to go to LMS, apart from the challenges, in addition to it, is it had been I did last year, I wouldn't have those videos still on the YouTube channel. For some of you that find this one also not clear, they can easily go and watch this one and watch that one as well, so that you have enough resources so that you can be able to take or to benefit most of what is expected of you. So like I said, I will also take this one there and I will share the link to you. When I share the link, you can go to the account and locate this. And if you need any other one similar to this, you can also have that of last year and you can compare the explanation. I'm very much sure when you compare the explanation, because I may not be saying exactly the same thing or the way I explain, I might find it easy to explain then maybe do 1 or 2 readings like that. So you can make use of that, and I'm very sure it will help you a lot. In the absence of any question, I think I will end the meeting now.
Hello, sir.
The material.
Go ahead.
Sir, can you give us the material, sir?
Okay,
All right.
but that
Thank
should
you,
be
sir.
at the end of the classes. Is that okay?
Yes, sir.
It's all right. So we thank you all for coming. I wish you all the best.
Yes, sir.
Thank you.
Thank you, sir.
Thank you, sir.
Yeah, you're welcome.
Thank you. Thank you.